Welcome back to our channel. Today we will discuss the potential mechanism through which antibiotics kill the bacterial cells. Join us as we explore the groundbreaking research that is reshaping our understanding of antibiotics and how they combat bacterial infections. For decades, antibiotics have been hailed as lifesavers, yet the precise mechanism by which they kill bacteria has long puzzled scientists. However, a recent study has unveiled a crucial piece of the puzzle. It reveals that antibiotics like penicillin unleash destructive molecules that fatally damage bacterial DNA, setting off a complex series of cellular events. This newfound understanding could be a game-changer for researchers developing new antibiotics, particularly as many bacteria have evolved resistance to current treatments. Now a new study by MIT and Boston University researchers reveals the killing mechanism behind all three major classes of antibiotics. The drugs produce destructive molecules that fatally damage bacterial DNA through a long chain of cellular events. The researchers believe that understanding the details of this mechanism could help improve existing drugs. Over the past 40 years, few new antibiotics have been developed, and many bacteria have become resistant to current drugs. In 2007, it was discovered that three classes of antibiotics, quinolones, beta-lactams, and aminoglycosides, kill cells by generating highly destructive molecules called hydroxyl radicals. Initially, it was thought that these radicals indiscriminately attack any cell components they come into contact with. These hydroxy radicals can oxidize proteins, they can oxidize DNA. However, most of this damage is not fatal. Hydroxyl-induced damage to guanine, one of the 4 nucleotide bases in DNA, proves fatal to bacteria. When this damaged guanine is incorporated into DNA, cells attempt to repair it. However, this repair process inadvertently accelerates their own death. While this mechanism doesn't explain all bacterial cell death, it accounts for a significant portion of it. Research into DNA repair enzymes suggested to scientists that oxidized guanine, the damaged form of guanine, could be involved in antibiotic-induced cell death. In their initial research phase, they demonstrated that a specific DNA copying enzyme called DNB, which is part of the cell's response system to DNA damage, is highly efficient at using oxidized guanine to synthesize DNA. The DNB enzyme inserts oxidized guanine not only opposite its correct base partner, cytosine, on the complementary strand during DNA replication, but also opposite its incorrect partner, adenine. The researchers observed that when an excessive number of oxidized guanines were integrated into new DNA strands, the cell's attempts to remove these lesions were unsuccessful, leading to cell death. Once oxidized guanine caused by antibiotic treatment is inserted into DNA, a cellular repair system is activated. Specialized enzymes, MuTY and MuTM, are involved in this repair process. They make cuts in the DNA to initiate repair mechanisms that normally help cells manage the presence of oxidized guanine in their DNA. However, this repair process is risky because it requires opening up the DNA double helix and severing one of its chains while replacing the incorrect base. If two such repairs occur close together on opposite DNA strands, it can result in a double-strand break, which is often fatal to the cell. This system, which is typically protective and accurate, can become detrimental, leading to cell death. The researchers, funded by the National Institutes of Health and the Howard Hughes Medical Institute, also discovered that aminoglycosides, a class of antibiotics, may cause cell death through an additional mechanism. In cells treated with these antibiotics, oxidized guanine is inserted into messenger RNA, leading to the production of incorrect proteins. These incorrect proteins then trigger more hydroxyl radical production, resulting in further damage to guanine. The researchers are now focused on advancing their understanding of how antibiotics kill cells. Thank you for watching.